Hi everyone, we are from Maravillosa channel. Today we will discuss and talk about our product, which is insulin. Introduction, I will introduce myself first. I am NURZAHIRAH SYAFIQAH binti ZAILAN, and there is MAHASOOKAH RAHMANIAH, NAJWA QHAIRUNNISA, CHEN and Muhammad SYAHMI as our group members. What is insulin?
The first place we're going to go is the isolation area. This is where we take the fermentation broth from another site, bring it down, and start to separate it.
It takes over 2 months to make a vial of insulin. It's a complicated manufacturing process.
This is where we make insulin. First is the insulin crystals.
Manufacturing, labor, research and development, regulatory fees, promotional expenses, insulin donations, and profits is only $122 a box.
Basically, insulin is produced by the pancreas, and it's an essential hormone that's used by our body to absorb glucose and utilize that in stored as fats or essential lipids. The reason why insulin is important, it is because it's used for more than just glucose control. It's also used for cholesterol metabolism. Actually allow us to convert cholesterol into various components of cholesterol, such as a good cholesterol and a bad cholesterol, which eventually are used by the body for essential metabolic processes. How human insulin is made? Do you know? I don't know, but let's find out. Human insulin is grown in the lab inside common bacteria, Escherichia coli, as known as E. coli. It is by far the most widely used type of bacterium, but yeast is also used. Researchers need the human protein that produce insulin. Manufacturers get this through an amino acid sequencing machine that synthesizes the DNA. Scientists make insulin by inserting a gene that codes for the insulin protein into either yeast or bacteria. These organisms become mini biofactories and start to spit out the protein, which can then be harvested and purified.
Assalamualaikum warahmatullahi wabarakatuh and Insulin Chapter. My name is NAJWA QHAIRUNNISA binti HUSIN with student ID PA21083. Now, what are the best example and you need to know about of genetic modification is how bacteria have been engineered genetically to produce human insulin. If you are type 1 diabetic, you cannot produce the human insulin. You have to inject it for the rest of your life. And if you choose instant from the beef or pig, it doesn't work well as a human insulin. So, how do you make enough human insulin from millions of diabetics? The answer is to get the gene that makes human insulin from a person's DNA, cut it out, and stick it into a bacteria, and the bacteria will read the gene as it is one of them, and they will manufacture the protein insulin as it is one of its own proteins. So you basically use this bacteria a bit like a slave to produce human insulin. The great thing is that once you have done it, the bacteria will produce millions of times passed on the generation every time because it reproduces asexually and very quickly. You can get a population of billions of bacteria or making human insulins by genetic modification. So, to go through the process in detail, step 1, remove a plasmid from bacteria. Cut it open with some restriction enzyme. Step 3, locate and remove human insulin gene using the same restriction enzyme. Then, you need to combine an insulin DNA with the plasmid. To do that, you need the ligase enzymes and they get combined. Step 5, the bacteria are then incubated with the new recombinant plasmid and they take it out. The bacteria will then take out the plasmid and start to produce the insulin. You can then get that back to reproduce and it will pass the gene on to all the other bacteria.
Hello everyone! First of all, did you know that animal insulin is the first type of insulin to be administered to humans for controlling diabetes? Moreover, it's derived from cows and pigs and used until 1980s. Due to research and development in science technology, recombinant DNA was introduced. The steps in producing insulin involve complementary DNA or cDNA and Escherichia coli. So, what is complementary DNA and how insulin are made from it? Let's find it out. Complementary DNA is used to clone eukaryotic genes in prokaryotic cells. In genetics, complementary DNA or cDNA is a DNA synthesized from mRNA template catalyzed by the enzyme reverse transcriptase and DNA polymerase in a process called process reverse transcription. What's more, cDNA is used in insulin production because it represents the expression of the genes without the introns, and there is no process of RNA splicing in bacteria cells. The production of insulin using cDNA started with plasmid DNA. Plasmid DNA is isolated from bacterial cell, which is E. coli. Then, mRNA that codes for insulin is extracted from the human pancreatic cell, to be more specific, the beta cells of the islet of Langerhans in pancreas. The second step is to cut and open the plasmid using restriction enzyme. Then the third step is to locate and remove human insulin gene using the same restriction enzyme. Next, the fourth step, we need to combine the insulin DNA with the plasmid using DNA ligase. Step 5, bacteria are incubated with the new recombinant plasmid. Then the bacteria will take up that plasmid and start to produce insulin in the fermentation tank. Then, that bacteria can get to reproduce and it will pass that gene on to all other bacteria, and this process is known as amplification. Moreover, insulin gene will be expressed via transcription and translation in clones of bacteria. Human insulin is extracted and purified from E. coli. And that's how insulin is produced. That's all.
Synthetic insulin is made by using recombinant DNA technology. Scientists insert gene code of insulin into the plasmid of bacteria to produce synthetic insulin. In fact, there are two types of usage of synthetic insulin, which are diabetic and non-diabetic. For diabetic applications, synthetic insulin can be used as a medication in treating high blood glucose. Synthetic insulin is able to treat diabetes mellitus type 1 and diabetes mellitus type 2. Synthetic insulin helps in transporting glucose from blood into other body tissues for generation of energy. Therefore, blood glucose level of diabetic patients can be kept within a target range. Besides, for non-diabetic applications, synthetic insulin can aid in wound healing, parenteral nutrition, anti-aging, and bodybuilding. Synthetic insulin can be categorized under three main groups, which are fast-acting, intermediate-acting, and long-acting insulin. Onset of action of an insulin is how quickly it acts. Peak of an insulin is time of the insulin at maximum strength, and the duration of an action of an insulin is how long time the insulin lasts. Rapid-acting insulin has onset of action of 15 minutes, peak effect at 1 hour, and duration of action of 2 to 4 hours. Besides, regular-acting insulin has onset of action of 30 minutes, its peak effect is at 2 to 3 hours, and duration of action of 3 to 6 hours. Furthermore, intermediate-acting insulin has onset of action of 2 to 4 hours, its peak effect is at 4 to 12 hours, and the duration of action is 12 to 18 hours. Apart from that, long-acting insulin has onset of action of 2 hours, it does not have peak effect, and its duration of action is up to 24 hours. Lastly, ultra-long-acting insulin has onset of action of 6 hours without peak effect, and its duration of action is 36 hours or longer.
Assalamualaikum. Saya Muhammad SYAHMI, nombor matrik PA21078. Saya akan membentangkan anything additional about E. coli. Most forms of E. coli are harmless and even can keep your digestive tract healthy, like the good bacteria from probiotics. But other strains of the bacteria can cause diarrhea or worse symptoms if you consume a food or drink that is contaminated with E. coli. We tend to associate E. coli with food poisoning, but you also can get pneumonia, experience trouble breathing, or even get a urinary tract infection. 75% to 95% of urinary tract infections are caused by this bacteria.
Thank you.